Hello again everyone, this is Dr. Habib, and today we'll be discussing the oxygen-hemoglobin dissociation curve. If you have any comments or questions, please leave them below, and I'll do my best to get back to you. In summary, we'll be discussing the structure of hemoglobin, followed by the causes of the right and left shift of the oxy-hemoglobin dissociation curve, a look at fetal hemoglobin, and finally a clinical correlation point, carbon monoxide poisoning. So let's first start by having a look at the structure of hemoglobin. So hemoglobin comprises of 4 globin chains. You have 2 alpha and 2 beta chains. And each chain is bound to a heme group, which is essentially one ion which is bound by a porphyrin ring, and this binds to one molecule of oxygen. Okay? And as we have 4 porphyrin rings, each molecule of hemoglobin can therefore bind 4 molecules of oxygen. So let me just roughly illustrate this. It's also important to remember that we have 2 types of states in which hemoglobin exists. One is the T state, also known as the 10 state, and the relaxed, meaning R-phased. What this basically means is the 10 state has a lower affinity for oxygen, and the relaxed state has a higher affinity for oxygen. Okay? Before we move on to studying the curve itself, let's have a look at a concept known as cooperative binding of oxygen. So let's say oxygen is going to bind to this subunit over here. Let's call them 1, 2, 3, and 4. So basically binding of the first oxygen molecule is going to be the most difficult. What this does is it leads to a conformational change in the shape of the other subunits, which will therefore make it a lot easier for subsequent 3 molecules of oxygen to bind. And once the second one is bound to oxygen, it then makes it easier for the third oxygen molecule to bind. And finally making the 4th molecule of oxygen easiest to bind. Therefore, any one molecule of hemoglobin has the highest affinity for oxygen once the first 3 subunits have already bound to oxygen. So let's have a look at the oxygen hemoglobin dissociation curve on the next slide. So we have the partial pressure of oxygen in the blood on the x-axis in millimeters of mercury, and we have the percentage saturation on the y-axis ranging from 0 to 100%. So let's say that the red line is the sigmoid shaped, which is a normal adult hemoglobin, and you will see it has a sigmoid shape, and the reason for this, as opposed to it being linear, is because of the concept of cooperative binding, which we looked at on the first slide. What this basically means is, as you will see in increasing amounts of partial pressures, the amount of percentage saturation doesn't really alter all that much. So even for large increases in partial pressures, the saturation has fairly plateaued out, or it's fairly fallen flat. Okay? Before we move on to the reasons for the right shift of the curve, let's have a look at something called P50. Now P50 is the partial pressure of oxygen in the blood, at which hemoglobin is 50% saturated. And you will see clearly why, with the right and left shifts of the curve, the value of P50 changes. In healthy adults, it's about 26.6 millimeters of mercury. So let's have a look at some of the causes for the right shift. So we have decreasing pH levels or increase in hydrogen concentration in the blood. We have high amounts of carbon dioxide, increasing temperature, and increasing amounts of 2,3-diphosphoglycerate. Let's have a look at them in a bit more detail. So why does low pH cause a right shift? Well, in an acidic environment, the binding of hydrogen ions is going to lead to a decreasing affinity of hemoglobin to oxygen. So the outcome of this is that you will require higher partial pressures of oxygen in order to increase the percentage saturations as we go up on the graph. So that's the difficult aspect of it. However, the beneficial aspect of this is with decreasing amounts of partial pressures, you will have more oxygen readily delivered to the tissues. Now, one important organ where this happens is skeletal muscle, where you have relative acidic environment in comparison to, say, the lungs, where oxygen is quite abundant. Having a look at increasing carbon dioxide levels. So we all know that carbon dioxide is a byproduct of aerobic respiration. Carbon dioxide is mostly present in the blood as carbonate ions, okay? And generation of bicarbonate ions is going to lead to a release of one hydrogen, okay? And this, similar to low pH, is going to decrease the affinity of the hemoglobin and cause a right shift of the curve. All right. And also, carbon dioxide is going to lead to formation of carbamino compounds and carbaminohemoglobin, and that will also decrease the affinity of hemoglobin to oxygen. Another thing to bear in mind is the outcome and the end product of having a low pH or an increase of carbon dioxide is that it stabilizes hemoglobin in the T state, the tense state, which basically means lower affinity. Having a look at the temperature, skeletal muscle is quite, warm and hot when you're exercising. It requires a lot of energy, and that will also shift the curve to the right. Why is that? Because increasing temperature can also denature the bond between the heme group and the oxygen. And lastly, having a look at 2,3-diphosphoglycerate. This is basically a byproduct of glycolysis, which happens in the red blood cell. And increasing 2,3-DPG amounts will also bind the hemoglobin molecule and cause a conformational shift, which then again reduces its affinity to oxygen. And on the other hand, we have all the opposite reasons, such as increasing pH, low amounts of carbon dioxide, and low temperature. These two are happening in your lungs because it's cooler, you breathe in air, and also you release a lot of carbon dioxide, exhale carbon dioxide into the air. And one place where you have low amounts of 2,3-DPG is fetal hemoglobin, which we'll come to next. So the fetal hemoglobin is going to look something like this. It's all the way to the left, all right? So you will see it has a really high affinity for oxygen, and because this difference is maintained, let's assume the one in the red is maternal hemoglobin or hemoglobin adult, and this is hemoglobin fetal. Because of this change or the difference, it can then uptake oxygen into its blood from the placenta. And finally, having a look at carbon monoxide, this equation here shows the formation of carboxyhemoglobin. Hemoglobin binds carbon monoxide with affinity about 200 times more compared to oxygen, okay? So the formation of carboxyhemoglobin is irreversible. And the concerning thing is that it does this at quite low partial pressures. Once carboxyhemoglobin is formed, the saturations of oxygen still appear normal. So this is quite misleading, because despite this, the body's tissues are suffering severe hypoxia as a result of carboxyhemoglobin poisoning, and this leads to a death if untreated. Treatment for this is high flow oxygen and even hyperbaric oxygen. So that concludes today's talk. I hope you found value in this. Please like the video and subscribe to the channel, and I'll see you soon in the next video. Thank you.